Can you see?
Yes, ma'am.
Yes, ma'am. Today we'll learn about lymph more detail. Then we'll learn activities, okay, the photosynthesis one. After that, everything will be over. Now let's start. We have seen here, right? There are many blood capillaries.
Yes, ma'am.
So this one, blood capillaries, we'll draw it here like this.
Excuse me, ma'am.
Hmm.
Ma'am, yesterday for the lung, ma'am, can you explain it just right once again?
What did you say? What about
I
what?
said that lungs one yesterday, ma'am, which you taught. Can you teach it once again? That
Lungs?
figure. Ma'am, can
I,
you?
lungs? No, I didn't teach lungs.
No, ma'am, that figure about that.
Tell me, what did I teach? Tell me.
Ma'am, I forgot the term about that one.
Lung? No, lung. I didn't teach lung.
No, wait. Sorry, you taught the excretory system.
Yeah. Yes.
Sorry, ma'am, I forgot.
Where is lung and where is excretory system?
No, sorry, you taught about excretory system and the transport of food and all in plants and
Yes.
excretion in human beings. That
Yes.
you by mistake I said lungs.
Okay. Okay. Let's. In that, what you need? Lungs, right?
Kidney.
Kidney. So the kidney. Functional unit of the kidney is nephron. Okay, and nephron is made up of Bowman's capsule and tubules.
Okay.
Okay, now this nephron, right? There are millions of nephron in kidney. Okay, and what happens in the single nephron? So let's see. In this Bowman's capsule, what do we have? We have glomerulus, right? It has many blood capillaries.
Okay, ma'am.
Yes, what, Ria? Then this cup-like structure is what we call as Bowman's capsule. Okay, and this thing, this tube-like structure, tubules. Okay, that is known as tubules. These all make what? Nephron. Okay, now here dirty blood will come in. Okay, through renal artery, dirty blood will come and it will go to the glomerulus. Okay, while passing through the glomerulus, right?
Yes, ma'am.
What will happen? Yeah. What will happen?
Ma'am, it will get filtered, ma'am. Glucose, carbohydrate, ma'am, it will be
filtered. Yeah, and it will get collected in the Bowman's capsule. Okay, these things will be filtered in the glomerulus. Okay, and what will happen?
Can you ask once again?
I asked all these things, right? Will be filtered in the glomerulus. Then it will be collected in the Bowman's capsule. Okay. Now, what will happen here? Now, filtration work is over. Now, there will be what? Reabsorption. Right? So, the waste material will be
sent back.
Will be left in the tubules. And the useful material will be carried by the blood capillaries, will be reabsorbed by the blood capillaries. Okay, this glucose, amino acid, and some salt which are useful. The rest of them will be left in the tubules, right? Many excess water, then salts and urea. These all things will form what? Urine. Okay, then it will go to the urine collecting duct in the urethra. Okay, then urinary bladder, then to urethra, it will be excreted out. Okay,
ma'am, before urinary, ma'am, before urinary bladder, ma'am, what it is written?
Ureter.
Okay.
This tube-like structure here, we have seen here, this one. Okay, we have two ureters. Understood? So millions of these nephron will be working together and they will form urine. Okay. Now, any other doubt here? No, right?
No, ma'am, now got it.
And in transport of food?
Ma'am, this is easy.
Easy, right? Okay. Yes. Now, what were we? We were in the lymph. So, this is our blood capillaries. Okay. Many other materials are here. And this is what? This is cells. Okay. These all are cells. And this is blood capillaries. Okay. Now, this blood capillaries will be having pores, small holes. Okay. You can see here, holes are present, right?
Yes, ma'am.
Now, see. So, this plasma and all, what will happen? This plasma and all will come out in the spaces between the cells. And all the plasma, the proteins, the cells, or other things will come out and they will form what? Lymph. They will form lymph. So what will happen to this lymph? So here there will be, this is lymph capillary. This plasma here will flow into the lymph capillaries, because this plasma, this lymph, right, they don't contain red blood cell, RBC. So they are what? Colorless. Okay, it will flow inside. Now you will ask how? How come? Because there is no space here, nothing is here, and it is closed. Then how come this lymph will flow inside this blood lymph capillaries? Now let's look.
Now, ma'am.
Let's see here. The lymph capillaries are like this. Okay, when the lymph has to come in, what will happen? Okay, so through this it will turn. Okay, so lymph will come inside. When it comes inside, what will happen? The pressure will be created. So this again, it will be changed into this shape. So it won't allow this lymph to come out. Only one way valve. Okay, one way door. Understood?
Yes, ma'am.
So this is wherever there is capillaries present, right? Lymph will be present. Any part of our body, lymph will be present. See, this all are lymph. You can see this all things are lymph. Okay, and there will be what? This dot structure you see here, right? This dot structure. What is that? That is checkpoint. Or you can say security point, and it is known as nodes. What does it do? If any pathogens comes, right, this will act as police. Okay, it will kill off. Okay, this lymph nodes, and this lymph capillaries will go to lymph nodes, right, middle point, and then they will go to larger vessels, lymph vessels. Lymph capillaries is small. Okay, larger lymph vessels they'll go. Then they'll go back to the veins. This is the flow of the lymph. Okay, understood?
Yes, ma'am.
So whenever sometimes when you get up, right, what will happen? You will see the swollen eye, right? That is because of lymph. Lymph will be gathered in outside of this cell, so it will cause the swelling. Or you can say, if there is some swelling, somewhere swollen, there is chance that there is infection in your lymph. Understood? Everyone got it? So blood capillaries, from there lymph will come out, and then from there lymphatic capillaries, then larger lymph vessels, then veins. Any doubt?
No, ma'am.
No, right? You got right? What is the lymph?
Yes.
Its work? Its work is three things. One, it will clean the blood. Okay. Second, what it will do? It will fight pathogen. Third, it will carry the fat substance. Okay.
Okay, ma'am.
That's about the lymph.
Ma'am, can you repeat the first one?
Clear blood. Okay. Second, fight pathogens. Third, absorb fats. Main function. Other functions are also there, but this are the main function. Okay. This blood vessels will be carrying the dead cells and all, right? So it will clean that too. Okay. So Shazan and Riya and Shashank.
Yes, ma'am. Understood.
Understood, right? Okay. Next, we'll see activities. The chlorophyll that we have seen, right? Photosynthesis. I have not taught the activities. So 3 activities we'll do. First is the sunlight is necessary for photosynthesis or not. Based on that activities. First thing, what we'll do is we'll take the leaf and we'll put the leaf in dark place, okay, where very dark place, where the leaf won't get sunlight for 3 days. Why do we do that? That is called de-starch. The starch content in the leaf will be used up. Okay, that's why we are putting it in a dark place for 3 days. Okay. Next, what we'll do? We'll take out the leaves and we'll put that plant in the sunlight. And before that, what we'll do? We'll take the leaf, one of the leaf, and what do? We will cover with aluminum foil, okay, middle portion, small portion, okay. So here the sunlight will not enter, and all the rest of the leaf part, sunlight will enter. This is the thing we are doing. Then we'll keep this thing in sunlight for 3 or 4 days. Then we'll plug that leaf and put it in water. Why do we do that? We'll break the plasma membrane, the cell membrane, so that it will be permeable to iodine solution. why do we use iodine solution? Come on. Why do we use iodine solution?
Ma'am, I forgot. I knew this.
Everyone know. Shashank, can you tell? Yes or no? Riya?
Yes, sir.
Yes.
To prevent chlorophyll.
See, if we put iodine solution in a leaf, what will happen if there is blue-black color? That means starch is present. Okay, starch is what stored form of the food in the plant. Okay, so and after that, what we will do before putting the iodine solution, we will take the leaf and we'll take a beaker. Okay, and we'll take a beaker and we'll put alcohol in that, and then we'll take another bigger beaker. Inside that we'll pour water. Okay, then we'll boil it. Why we are not putting the alcohol and water together? Because the alcohol is highly inflammable. That means it catches fire quickly. So that's why we have to boil this leaf in alcohol. Okay, and so we'll put the water here. Okay, and then there will be what? Alcohol. Directly we can't boil, right? Because it is highly inflammable. That's why we are using water. So when we heat, this water will boil, and here this beaker will also boil. So alcohol will boil, and this leaf will also boil. Okay, what will happen? The leaf will get decolorized. Okay, and we'll put this leaf in the petri dish container. Then what we will do? We'll put iodine solution. What we will find is that wherever there was this slip, right, aluminum foil, there was no blue-black color. Okay, starch was not present, and wherever there was no slip, okay, where sunlight was falling, what did happen? Starch was present. So what we will conclude? We'll conclude that sunlight is necessary for photosynthesis. Okay, okay or not? Got it? Shazan? Yes,
Yes,
sir.
ma'am.
What did you get?
That when it is covered with ammonium foil and ma'am, it is kept in iodine solution inside the water, and the water is boiled. So ma'am, where the ammonium foil is covered, ma'am, there will wait, I'll tell you. Ma'am, where the ammonium foil is covered, ma'am, that place will not become blue or black, and the other will become blue or black because starch is present in that.
So starch is present. What it will indicate?
Ma'am, that food is stored.
Yeah, a leaf made food. Okay,
Yes.
with the help of sunlight. But here in this case, food is not made because because sunlight was not present. Okay, no blue-black color, no starch formation. So we can conclude that sunlight is necessary for photosynthesis. Next, we'll see chlorophyll is necessary for photosynthesis. Now, see here, we take leaf. Okay, variated leaf. That means some parts there will be green and some parts there won't be green color. Okay, irregular. So that type of leaves is called variated leaves. Okay, we'll keep this this leaf for in the dark place for 3 days. Okay, then in sunlight for 3 days. Then same step we'll do. What step? Decolorization, boiling in the alcohol. How do we boil in alcohol? We use water bath. That is, we put water in a container. In another container, we take, what do we take? The alcohol and the leaf. Then we boil it. Not directly boil. That is water bath. Okay. Then leaf will get decolorized. Then what we will do? We'll put iodine solution. What we will find? Wherever there was green part, right? That part had what? That part gave blue-black color. That part had starch. Okay. So we can conclude wherever there was chlorophyll, food was formed. That means chlorophyll is necessary for photosynthesis. Understood? Understood or not? Yes, ma'am. Shazan?
No, ma'am.
No.
No.
So here we have leaf, variated leaf. Okay. In this leaf, some part will be having green color. The
Okay.
other part will not be having green color.
Okay, ma'am.
That is called variated leaf.
Okay.
And what we will do, we'll put this leaf in dark place for 3 days, and then we'll put this leaf in sunlight for 3 days.
Okay.
Why do we put it in dark place? To de-starch it. That means whatever there was
Stored food means that
content,
can be used.
yeah, it will be getting used up. Then we put it in sunlight for 3 to 4 days.
Okay.
Then what we will do? We'll boil
In alcohol.
leaf, okay, in alcohol. How we will boil?
Ma'am,
This.
why we arrange that like that?
Like this. Okay. Directly we cannot boil alcohol.
Okay.
Okay.
It will
It
catch
is
fire.
very highly inflammable.
Yes, ma'am.
It will catch fire. That's why we don't do that. Okay. Then after this, what will happen? All the chlorophyll will be removed. Okay. And what will happen? No chlorophyll present. Okay. Next, what we will do? We'll put iodine solution. Wherever there was chlorophyll, there we find starch present, blue, black color.
Okay, ma'am.
Why we are doing this alcohol? Why we are removing this chlorophyll? Because, right, in this case also we removed chlorophyll, right? Why?
Check the starch.
No, because no, we are putting this iodine solution, right? It will interrupt. We cannot
Okay.
see properly this chlorophyll and this blue-black color. We will not be able to differentiate properly.
Okay.
Okay, that's why we are removing the chlorophyll,
Okay, ma'am.
decolorizing the leaf.
Okay.
Okay, so we
Ma'am,
can find,
it is essential to remove chlorophyll before this.
yes.
Okay, ma'am.
Okay, we find wherever there was chlorophyll present, there only food was formed. There only we got blue-black color.
Okay, ma'am.
Next experiment is what? Chlorophyll is necessary for photosynthesis. First step again we'll do the de-starch. What we will do? We'll take the plant and put it in dark place for 3 days. Then after that, what we will do? This is the plant. We'll take one jar and this is the cork. Some part of the leaf we'll put it inside. And the other part we will be leaving it outside. Okay. And inside, what will happen? There will be one solution present, that is potassium hydroxide solution. What does this do? Potassium hydroxide, it absorbs KOH. Okay, it absorbs carbon dioxide. And here outside here in the leaf, the carbon dioxide are available in the atmosphere. Here there is no carbon dioxide. Okay. After that, what we will do? Again, we will decolorize the leaf. Okay. Then we will pour. What we will pour? Iodine solution. We will find wherever the outside part of the leaf. Okay. That part was green. It's a blue-black color. Okay. That part was blue-black color. And so here there was starch present, but here in this case, here this part, which was the part of this which was inside the jar, there was no blue-black color. The food was not formed. So we can conclude that carbon dioxide is necessary for photosynthesis. Okay? Okay or not? Got it?
Yes, ma'am.
Shazan, any topic? Okay, then pass over. Bye-bye. When will you complete this PPT? Timing, Wednesday. Okay. And on Monday, I think Monday, Tuesday or Wednesday, there will be this LFP test. Okay. Because this chapter is over, you will be having test.
And next week?
Yeah, next week.
Okay, ma'am.